This is part 2 of the lecture on cell structure and function. And the last slide that, well, this is the slide that we stopped on in the last lecture. And just a review, we learned that the plasma membrane is composed of lipids, proteins, and carbohydrates, 3 of our macromolecules that we learned about in chapter 2. So the lipid portion of the plasma membrane includes phospholipids, a phospholipid bilayer, and cholesterol, which makes the membrane less fluid. Lipid-soluble materials easily cross through the phospholipid bilayer. So what are lipid-soluble materials? Other lipids and gases, small molecules like oxygen and carbon dioxide, easily cross through the phospholipid bilayer. But ions, which are atoms with a charge, so these are your electrolytes like sodium and calcium and potassium, and water-soluble compounds like glucose and amino acids. These do not cross the phospholipid bilayer, so they must travel through the plasma membrane by a channel protein or a carrier protein. The phospholipid bilayer is composed of a hydrophilic head. It contains a phosphate group, and each hydrophilic head faces the outside or the inside of the cell, and the hydrophobic tails face each other, and they are composed of lipids. The proteins, the large blue molecules that we saw scattered throughout the plasma membrane, have different functions, but some are transmembrane proteins that span the width of the membrane. In other words, this is a transmembrane protein. Others are loosely bound either to the surface or partly embedded in the phospholipid bilayer. Some can drift and others remain in position, and the composition of the membranes can change over time. There are 6 types of membrane proteins. There are receptor proteins. There are channel and carrier proteins to help non-lipid soluble molecules to travel across the plasma membrane. Ions and water-soluble molecules have to travel through channel proteins and carrier proteins. Enzymes, which speed up chemical reactions, anchors, and identifiers. This table gives you the function of the different proteins and an example of each one. Receptor proteins are sensitive to specific materials that bind to them. So they bind to certain chemicals, and that creates a change in the cell's activity. An example is the binding of insulin to membrane receptors in our muscles and our liver, cause those cells to absorb glucose from our blood and lower our blood glucose levels. Channel proteins form a pore or channel that permit water-soluble or charged particles to pass through. For example, there's a calcium ion channels that are crucial to muscle contraction. Carrier proteins are the same as channel proteins, but they chemically bind to the substance they are transporting and then move it across the plasma membrane. Enzymes catalyze reactions. Anchoring proteins attach the plasma membrane to other structures inside and outside of the cell. And recognition proteins or identifiers identify a cell as self or non-self, normal or abnormal, to our immune system. So when we're babies, one thing that's happening in our immune system is it is learning which chemicals are found on the cells of our body. And those cells are called self cells, so that they can destroy the non-self cells that come into the body. One group of recognition proteins is the MHC, major histocompatibility complex, which is the tissue compatible molecules. They have to be matched up if a person is getting an organ transplant or tissue transplant. Carbohydrates on the surface of the plasma membrane either combine with lipids to form glycolipids, or they combine with proteins to form glycoproteins. They can function as lubricants, adhesives, receptors, and part of the immune system self-recognition. List the general functions of the plasma membrane. Well, it surrounds the cell and separates it from the environment, and it allows certain materials to pass through it. The component of the plasma membrane that is primarily responsible for forming a physical barrier between the cell's internal and external environments is the phospholipid bilayer. Which functional class of membrane protein allows water and small ions to cross the plasma membrane? And that is a channel or a carrier protein, but I think that this is a channel protein. All right, permeability is the ability of the cell's plasma membrane to transport substances across. Our cells are selectively permeable. That means they allow some substances to cross and prevent others. So our plasma membranes are selectively permeable. Movement across the membrane is either passive or active. If the movement is passive, it does not require energy in the form of ATP. Examples of passive transport are diffusion, osmosis, and facilitated diffusion. Active processes require energy, usually from ATP, and examples are active transport and vesicular transport, called endo and exocytosis. And carrier-mediated transport methods can be active or passive, but they involve a carrier protein. Diffusion is defined as movement of a molecule from high concentration to low concentration. The difference between the concentrations is called the concentration gradient. The concentration gradient is simply a concentration difference. So if this is the cell, maybe I'll have a picture. I don't like that one. This is diffusion across the plasma membrane. This is just showing you diffusion of dye in a beaker of water, but we're actually focusing on diffusion across the plasma membrane. Now, some molecules can cross through the phospholipid bilayer. Molecules like lipid-soluble drugs, alcohol. It's why it one of the reasons why it has the effects of alcohol are so quick. Fatty acids, steroids, and oxygen and carbon dioxide. These are substances that can easily cross through or diffuse through the phospholipid bilayer. And then some substances pass through channel proteins. If they are large substances, they may require a carrier protein. So what you're looking at here is the diffusion of water and small particles across the membrane. It looks to me right now as if this is water. If the concentration of water on the outside is high and the concentration on the inside is low, if you look at the number of water molecules, So at this point in time, more water is going to move into the cell from high to low. And this molecule is cholesterol. And cholesterol is lipid soluble, so it can diffuse through the phospholipid bilayer. If its concentration is high outside and low inside, it will diffuse into the cell. These molecules are not going to move through the phospholipid bilayer because they are large and they are not lipid soluble. They are water soluble molecules. Osmosis is a special type of diffusion. It is the diffusion of water. It occurs across a selectively permeable membrane, which our plasma membrane is. It's freely permeable to water, but not to some solutes. So water will flow from the low solute concentration to the high solute concentration. When we talk about the urinary system, I always tell students water follows salt. Water moves where you have the highest salt concentration. But for now, you can say that water follows solute. Water will move to where the solute concentration is highest. Osmotic pressure is the pressure produced by the concentration gradient across the plasma membrane. And it's going to be a higher pressure the greater the difference in concentration across the membrane. This is an example of osmosis. This is called a U-tube. It's actually a U-tube, U-shaped tube. There's side A and side B, and then there is a selectively permeable membrane at the very bottom of it. This membrane is allowing water to pass through, but it's not allowing those large purple molecules to pass through. So we know water follows solute, and there's more solute here. So water is going to move to side B. And the pressure that is created by applying a force on side B, that's called hydrostatic pressure. Tonicity is the effect of different solute concentrations on the shape or tension of the plasma membrane. If a solution is isotonic, it doesn't cause a net movement of water into or out of the cell. So water molecules move in and out at an equal rate. So the cell will stay the same size. It will retain its normal appearance or remain the same size in an isotonic solution. 0.85% salt, sodium chloride, is the same thing as what we term in medicine normal saline, which is used as IV fluid sometimes. If it were just water, it would cause our cells to swell and burst. And if it had too much salt in it, it would cause our cells to shrink, our red blood cells to shrink. So the solution has to be isotonic to our fluid in our bodies, our body fluid. Okay, hypotonic solutions have a lower solute concentration than inside the cell. So if a cell is in a hypotonic solution, it will gain water. So for example, we know inside the cell is 0.85% salt. Sorry about that line. So let's just say 0.85% salt. Let's just say outside was distilled water, 0% water. So the concentration of solute is higher in the cell. Water is going to move in. Sometimes that can cause the cell to burst or lyse. If it happens in red blood cells, we call that hemolysis. Hypertonic solution means the cell is placed in a solution with a higher solute concentration. It will cause the cell to lose water, and the cell will shrivel and dehydrate. In red blood cells, this is called crenation. Example, if you place cells in a solution of 5% sodium chloride, that would be hypertonic.